All right. Good morning, good afternoon, everyone. This is Scott Mushaking with Eurofins, bringing you our webinar today in collaboration with Rheonix that we have titled, Quickly Evaluate Your Environmental Monitoring Program, a new way to target recurring listeria in your facility. First, a little bit on our guests. Today we have Rheonix with us. Rheonix offers highly multiplexed assays for use in food and beverage testing, clinical diagnostics, and research. The Rheonix and Compass workstations and assays provide fully automated molecular testing with a simple, intuitive workflow and a high level of accuracy. In our conversation today, we aim to cover an overview of Listeria environmental and testing concerns within a processing facility, pack house, or wherever your environment may be, highlighting the new technology offered by Rheonix and the Listeria PatternAlert. So I'd like to start by giving a brief introduction to our featured guest. We have Dr. Dan DeMarco. He's a globally recognized expert in food pathogen diagnostics with over 20 years experience developing and using rapid pathogen testing methods in R&D roles with major rapid test kit manufacturers and third-party testing labs. So as part of a Eurofins R&D group, Dan has direct involvement in evaluation of testing tools and as an advisor in their use and application across the Eurofins network. So good morning, Dan. In this webinar, Dan will discuss the challenges we face in environmental monitoring for Listeria and provide an overview of considerations for strain typing and molecular confirmation. Next, we have Megan Mern. She's the business unit manager of the Eurofins Microbiology Lab in Mounds View, Minnesota. Megan and her team perform various cultural and rapid detection testing methods, including the Listeria PatternAlert assay. So Megan today is going to be pulling from her experiences and build a little context to the application of the Listeria PatternAlert through a series of case studies. And then finally, from Rheonix, we have William Patram. He's the business and sales director for Rheonix. So as a leader of customer engagement activities with Rheonix, Bill will detail the technology and process flow of the Listeria PatternAlert assay. So welcome, Bill. All right, so before we begin, I want to relay just a few general notes on the webinar structure. Scrolling through on the wrong side here. The webinar is being recorded. Slides from the presentation as well as the recording will be emailed out to everyone registered within 3 business days, and we'll also post the webinar online for on-demand viewing. So during the webinar, you can submit questions at any point in time. There's a little sidebar menu on the GoToWebinar screen. All you have to do is select the questions tab, type in your question, and hit the enter key on your keyboard. Remember, you can submit questions throughout the webinar. At the end, we'll have a short Q&A session where all of the panelists will be able to provide answers to your questions. All right, so with that, Dan, I'm going to hand this over to you and let you get started.
Thank you, Scott. Appreciate that. Good morning, everybody, and thank you so much for joining. So for those of you who know me or seen me give a talk before, a presentation before, you know, I typically like to start with something maybe just a little bit humorous or a little bit lighthearted, just to sort of relax the audience, but also, you know, more importantly, to relax myself. So what I thought I would do today. So today will be no different, and I'm going to talk about a topic that's actually, I think, pretty appropriate for today's talk, which is Listeria. And I'm going to talk about what I consider my most important contribution to microbiology. And it's not actually a scientific contribution. It's in fact a single joke. And the joke, my hope is that someday this will enter the pantheon of great microbiology jokes as one of the greatest microbiology jokes of all time, and alongside such classics as What Did the Joker Say to the Spore-Forming Bacilli? And of course, that's why it's so serious. So this joke, which I think, again, could be as good as this one. And I know it isn't original, is what do they call a pathogenic Listeria in Mexico? I actually live in Mexico, so this was very appropriate for myself. And actually they call it El Mono. So there you go, my most important contribution to microbiology. All right, so with that, we'll move into the more serious stuff, and I'll start with just one introductory slide about Listeria and public health. So Listeria first came to prominence really in 1980s. In 1981, in Nova Scotia, Canada, there was an outbreak involving 34 pregnant women and 7 adults. They found El Mono in unopened packages of coleslaw, and they suspect the source was sheep manure, actually, that was used as fertilizer. As of 2021, so just 2 years ago, there's over 26 species formally recognized as Listeria. There's really only 6 that are viewed as important by the regulatory agencies, and have been the main focus of diagnostic test development. 2 of those are considered pathogenic. That's Listeria monocytogenes, of course, and then Listeria ivanhoe as well, which is mostly affects animals. The CD estimates there's about 1,600 cases of listeriosis each year and about 260 deaths. Compared to some of the other major foodborne pathogens, it's really not that infectious, but it is highly deadly. And it primarily affects infants, pregnant women, the elderly, and people with weakened immune systems, so highly vulnerable populations. So it tends to elicit a lot of sympathy and gets a disproportionate amount of attention, I think, because of that. I'm going to switch gears now and talk about environmental monitoring for Listeria species. So if you look at EMPs, they're really one of what we call the 4 pillars of food safety. So you have good manufacturing processes, of course, GMPs. You have your standards, sanitation standard operating procedures, your HACCP plan, your HACCPs, and then your EMPs. And the main objectives of environmental monitoring programs are basically to find pathogens or allergens in your environment before they can contaminate products. And they're also used to assess the effectiveness of basically your sanitation programs and your employee hygiene practices. And to be effective, an important point that sometimes gets forgotten is that it really has to include, in addition to having the program, you've got to look at the results and you've got to do root cause analysis when you do have a positive environmental hit. And you have to implement corrective actions as well. So just gathering the data is not enough. Even just gathering and analyzing the data is not enough. You actually have to do something with that data and implement some sort of corrective actions. So why do we want to monitor the environment? There's many reasons, but there's one big reason, and that's the fact that recalls are so expensive. This is an example of a company, I'm not going to name the company, but from 2018, this was a co-packer that produces a wide range of meat-based products. They had a positive environmental result for Listeria monocytogenes on just one production line. They did some additional testing by themselves and FDA as well, and they confirmed that multiple products manufactured on that particular line had become contaminated. So they stopped all production, and there was a recall initiated, 600,000 pounds of product. That encompasses roughly 30 different individual brands. And the contamination was eventually traced to a filling tube that was used on that particular production line. But if you look at the costs associated with this, the breakdown of the financial losses, it's really quite large. So the BI loss based on gross sales, 1.3 million, recall replacement cost, 2 million, increased cost of working, 60,000, inventory not shipped, 330,000, customer recalling fees, did not even know that was a thing, but 200,000 of those, additional slotting fees, whatever those might be, $5,000, and a PR consultant, of course, you need to have one of those, and then brand rehabilitation. And so all in, a total financial loss of $4.6 million here. This is a recall. This is not even an outbreak. So nobody got sick. This is just a recall. So this is sort of a best case scenario, if you will. Huge financial losses. And this is actually somewhat small compared to some others I've seen, particularly with salmonella. So the numbers, the costs are really staggering. So transient and persistent strains are really important to understand if you're talking about or thinking about environmental monitoring programs. Transient strains are those that enter into your facility but do not permanently reside there. So they're typically they can't adapt and replicate in the environment. And your standard control programs, your standard sanitation programs can eliminate them pretty easily. And they're only found occasionally during your routine surveillance. And they're considered lower risk because they're less likely to cause a large-scale contamination of product. But the persistent strains are the ones that we really worry about. And these are the ones that get into your facility and they establish permanent residency. And they can persist for months to many, many years. And your control programs just can't get rid of it. They can't eliminate it. They establish some sort of a niche in a harborage location. Usually that's in some sort of a biofilm. And they're much higher risk because you can cause these do cause contamination of product and can. And they are frequently found during routine surveillance. But an important point here is that your standard screening methods, your screening assays for Listeria, do not identify or differentiate between what's a transient versus a persistent strain. And there is an increased focus, there has been for many years actually now, by FDA on finding these persistent Listeria strains. What they've been doing is going to facilities and conducting swabathons, basically taking hundreds of swabs, recovering any Listeria that they recover, they then sequence. And this can create a risk for food manufacturers. Which brings me to strain typing and source tracking, which is a good way to differentiate persistent and transient strains. So what is source tracking or strain tracking? Basically, these are programs that are designed to find those physical locations of those harborage points where pathogens or sometimes even spoilage microorganisms can be found. And they can help you to determine the source, obviously source tracking of contamination when a positive product sample is identified. They typically use some sort of a isolate typing is how you actually differentiate the source. And there's a whole bunch of methods that are used. PFGE, of course, is a big one. Ribotyping, RET-PCR, standard traditional serotyping. There's different types of target sequencing that can be done, different types of whole genome sequencing as well. almost all of these methods require the isolation of the organism as a colony on a plate prior to doing this sort of that type of analysis. And they are generally very labor intensive and time consuming. And these systems that are not as labor intensive or time consuming because they've been fully automated, the Hygenia Rotoprinter and the Biodiversity Labs, are both being discontinued. So, I can get this to forward here. Which brings us to the Rheonix Listeria PatternAlert assay. So this system allows us to enable a rapid ID of recurring Listeria patterns across locations and time, and it works directly from a positive enrichment. So you don't need the isolate that's required of all those other methods I just showed. It's a fully automated process, which creates actionable results in about 7 hours. You do get a user-specific database of patterns. That's the output. Bill will talk a lot more about this when he talks in his section at the end, but there's no match to any sort of public database. And it gives you rapid and actionable data, which is important to help you identify the root cause. If you have a recurring contamination, find those harborage sites and eliminate them and address any sort of emerging issues. So food producers, if you ask them, can you use Listeria strain information? Would you use Listeria strain typing information? And they say yes, and they say they value that information, and you can use it to do these things, to identify transient versus persistent strains. But it's not widely used. And why is that? Well, there's several obstacles. So the first is liability. So there's a concern about links to current, past, or future outbreaks, and the fact that you can get full speciation. With the Rheonix Listeria PatternAlert, you don't have that match. I mentioned already, there's no match to a public database, and you don't get speciation of patterns unless you do have the option to run a Listeria monocytogenesis specific kit if you do want that information. The cost, of course, in food manufacturing, as we know, the pressure is there always to keep costs low, and that's across all departments. And if you look at the Rheonix system platform, it's half the cost of the standard strain typing methods that I just mentioned. Turnaround time is another big obstacle. 1 to 2 weeks at best case for some of these from an isolate-based method. And with the Rheonix system, we're talking about 7 hours post-enrichment, so a huge, huge advantage in turnaround time there. And then the resource requirements to run many of those strain typing methods I mentioned require significant laboratory infrastructure, people, highly technically trained personnel. And the Rheonix system is very low labor. The training and infrastructure requirements are low. It's a fully automated platform. So looking more closely at the turnaround time and the time to result comparison, if you look at current methods for confirmation and strain typing, as I mentioned, they can take up to 2 weeks. Basically, this kind of figure here is showing kind of the standard paradigm of food testing, where you have a sample enrichment, which can take between 1 and 2 days, and then you have your screening method, and these are typically in the 1 to 4 hour kind of time frame. And then you do a confirmation, and that confirmation is almost always a cultural-based confirmation, and that can take 2 to 3 days. So, and then if you want to add on the back of the end of that, your strain typing method, it's an additional 1 to 7 days. So you're talking about all in about 3 to 10 days. In contrast with the Rheonix platform, the sample enrichment does not change, so that's the same. The pathogen screening method that's up front does not change. It's everything on the sort of back end is where the changes come in. And you take the enrichment and you run that basically directly for both confirmation and strain typing. And it takes only about 7 hours. All right, which brings me to the topic of confirmations, and I did want to dive into this a little bit more because I think it's pretty important. It's also something that I personally am fairly excited about. So the Rheonix platform as a confirmation tool. So first, I need to start by mentioning that this is a new application. I think it has great potential. It is something we are exploring at Eurofins, but it is what I would consider a bonus feature of the system. So it's not been a focus of Rheonix during the method development for the PatternAlert assay, and the system is not really being marketed as a confirmation tool. But I view it as a potential game changer, and I think it could have a positive impact on all of food pathogen diagnostics. And I think this allows us the opportunity to influence the acceptance of direct from enrichment molecular confirmation approaches in food testing. And it allows you to confirm potentially any presumptive rapid screening method directly from that enrichment in about 7 hours, as I mentioned several times now. So why do we want to use molecular confirmation? Well, there's several reasons, but one of the main ones is that there have been ongoing issues with the cultural confirmations that we're using in the testing laboratories. They're slow, they're labor intensive, they're costly. They are the primary driver of results delays and bottlenecks in third-party testing labs today. Certainly in Eurofins, and I don't think I can speak for the other laboratories, but I'm pretty sure it's somewhat similar there as well. And they're a big driver of overall testing costs. And then of course we've had in recent years this issue of non-confirming presumptives. And there's some thinking that cultural methods may just not be quite as sensitive as these rapid screening methods that are being used. There's the detection of dead non-culturable cells that can complicate things. There's some of these difficult to culture or unique strains that are out there. And really, if you look at Listeria, if you look at the testing world, Listeria species testing, particularly environmental Listeria species testing, has been the most significantly impacted by this problem of NCPs. And so with this Rheonix system, or really any sequencing-based approach, with a species fingerprint is generated at the same time as a confirmation result, and can be used for strain tracking or for ID if you're using other sequencing-based approaches besides Rheonix. So why not molecular confirmation? If it has all those advantages and all those good things about it, why are people not using it? Why are we not doing this today? Well, there's several reasons, but one of the primary ones is that all rapid methods today were initially validated or certified by comparing them against the reference cultural methods, and culture is still considered the gold standard. Therefore, the answer that culture gives is always viewed as the, quote, correct answer. And there's been a concern about false negative results. What happens if you use a less sensitive method to, quote, confirm a more sensitive method? So a good example of this would be if you used an immunoassay to confirm a presumptive PCR result. And that's fine if you have a presumptive PCR and the immunoassay is also positive, but what if it's not? What if that is negative? Does that mean the target wasn't really there, or was maybe the immunoassay just not sensitive enough to see it? So can we, as I'm saying, we as a testing lab release a negative result in a case like that? And if you look at just even PCR methods of themselves, they have slightly different sensitivities, which depend on many different factors. So what if you just happen to choose a less sensitive PCR method and you select that as your confirmation method? So the question becomes, is the Rheonix PCR method, which this system is based on PCR, less sensitive than the rapid screening method PCR it's being used to confirm? And I think we don't necessarily know the answer to that question. One thing I would say, and there's a couple things I would say at this point, is that there is a lower potential impact from a potentially non-confirmed result, so-called false negative result with Listeria environmental testing, because we're talking about indicator testing and not pathogen testing. But it's also possible to argue that the situation I'm talking about here is what we face currently with culture, where we're using a slightly less sensitive method to confirm a more sensitive method. The other argument here is that, well, regulators need a colony on a plate to do these traceback or outbreak investigations, which is true, and that more importantly, perhaps lawyers need a colony on a plate in order to prosecute, and that is also, we believe, to be true. But the point I would make here is that 99.99% and even probably even a higher number than that of presumptive results are not ever tied to or implicated in any kind of a recall or outbreak investigation. And also logistically, by the time something like that would happen, most testing labs would have long since disposed of the cultures, and they would not be available for use in any case. All right, so I just have, I think, just one last slide here, and one of the things I struggled with as I was pulling this presentation together was trying to remain somewhat neutral and sort of as unbiased as I could. I am a big fan of the technology, I'll freely admit that, but I did want to try to be as unbiased as I could. So I said I'll put together a sort of a pros and cons list and then present that. But as I put that list together, one of the things that struck me was that the pros and cons ended up being basically the same thing, and it really had a lot to do with your point of view and your particular situation, whether you viewed a given attribute of the platform as a pro or a con. So what I'm showing here with this table is several attributes of the method, and I'm talking about the upsides of that and also the downsides or drawbacks of that. So first attribute is that it is, yes, a molecular method, right? So the big upside from my perspective is that it's not culture, right? So that's a good thing, but it's also depending on your perspective, that might be a bad thing, that's not culture. Because culture is, as I mentioned, the gold standard, and all the validations and certifications are based on those cultural methods. So it's also much faster. Molecular confirmations, in my opinion, are the way of the future. But again, the downside of that is that there's a lack of wider acceptance as a viable option. And there's, of course, very much a lot of resistance to change. And then you get also the benefits of strain typing. But the downside of that is you don't have a colony on a plate, which might be required for some sort of regulatory action. It's PCR based. That's a good thing in my opinion. It's a time-tested, proven technology. It's been around forever, highly sensitive and specific, and it's robust. But the downside here is, again, depending on your point of view, do you want to confirm a PCR result with another PCR method that could be viewed as suboptimal? That's the same technology fundamentally. So there's a nucleic acid isolation and purification step with the Rheonix platform, and that may give improved performance in some matrices, which is great, but you have this, again, see above I say here about what the acceptance of molecular confirmation approaches. It has multiple different PCR targets, which are unlike those used in any of the major rapid screening methods. But it also, as a downside, might be slightly less sensitive than those screening methods. Okay, another attribute, it's not whole genome sequencing. In my opinion, again, this is a good thing, because you don't have a potential for L mono identification or any concerns potentially about data loss. But that also could be viewed as a downside or a drawback depending on your point of view, and you get less information overall. It's fully automated. A great thing, ideal for a high throughput lab environment, easy to use. You load it and walk away. But again, not all labs are high throughput, and there is a footprint here, a fairly large footprint. So not every lab is going to be able to place this system in their particular lab. They just won't have the space for it. Strain typing, another attribute. You can do strain typing here, which is a very powerful tool for tracking, trending, and remediating, as you'll see and as you've seen. But again, not everybody needs or wants strain tracking. And then cost, this is a big one here. So on the upside, it's definitely it's comparable to cultural confirmations with a biochemical or genetic ID to species level on the back end. And it's really a bargain when you compare it to the alternative strain typing technologies that are out there. But on the downside, it's more than cultural confirmation to genus level alone, which is what many people actually do today. And so with that, I'm going to turn things over to my colleague, Megan Murren, who's going to run you through a couple of case studies. Thank you.
Thank you, Dan, for laying that foundation. That was great. All right, so we're going to move into some Eurofins case studies, and we're going to show how this new approach can help your EMP program. How to advance here. Okay, so the Eurofins Microbiology Laboratory in Mountain View, Minnesota started working with the Rheonix team back in 2022, and we onboarded the method in early 2023. We are currently the only third-party testing lab to offer this assay, and we are a preferred partner of Rheonix. We onboarded the assay because we saw that this would add value to our clients, and we do believe that this is the way of the future. So some of the value that we were seeing when evaluating the method was that it can help to resolve persistent Listeria issues within your facilities. It can provide early visibility to recurring Listeria, and it can be used as a non-culture confirmatory method. In addition, the key features that we were looking at, as Dan discussed, it has the ability to process directly from those presumptive positive enrichments, and there's no need for an isolate. And the key feature that really is the deliverable is the PatternAlert software. So it creates a company-specific database of patterns. That really is the thing that is the deliverable piece, and it's not comparable to any public database. So before diving into this, we're going to discuss 3 different client case studies here. And so the information that we're presenting is 100% confidential to our clients, and we're kind of generalizing the names of the companies and the client data here. So we're going to discuss 3 case studies. We're generically naming those company X, company Y, and company Z. And I wanted to discuss the naming convention for the client-specific databases and how that information is going to look. So, for example, company X, sends us a number of Listeria presumptive enrichments. We process those on the Listeria PatternAlert, and the deliverable is going to be this client-specific database. Now, the patterns are going to come across as generically named patterns. So, for example, XCO1 for company X, XCO2, and XCO3 are 3 examples of how those Listeria patterns might come across. Now, that is just representing Listeria pattern 1, Listeria pattern 2, and Listeria pattern 3 from that specific company X. So the database naming convention is very generic. There's no Listeria species mentioned. It's just the generic pattern name of that Listeria for that company. And so company Y and company Z, as we discussed those as well, you'll see that those generic naming conventions are used. Okay, so for the first case study, we're going to discuss company X. Company X is a client that has their own internal microlaboratory. And so they were having some recurring Listeria presumptive results during their in-house testing throughout their facility and operations for about 14 months. They wanted to identify the root cause and eliminate the Listeria contamination. They had come to us. They knew that we were onboarding this Rheonix Listeria PatternAlert method, so they were saving up their presumptive enrichments in the refrigerator and then planning to send those to us. So eventually, all of those presumptive enrichments were sent over to us for testing. And after analysis of 88 positive Listeria samples with the Listeria PatternAlert assay, we identified a primary pattern. So XCO2 was their primary pattern, for example, that was found to be causing these issues. So they were able to use the pattern name and the operational data with the floor plan to determine, where is this harborage site? Where is this coming from? And they eventually found that it was coming from a movable cart that was traveling throughout their facility, as you can kind of see on the floor plan here. This movable cart. So it was able to help them concentrate and focus their investigation to figuring out how to eliminate the source of that Listeria that was harboring in the cart. So essentially, it just helped them find the source, help them eliminate it, and eradicate that Listeria from their environment. So in our next example, we have company Y. So company Y was historically using cultural confirmation, followed by identification. So with that, they're determining, is it there, is it not there, is it confirmed, or is it not confirmed? They had seen an unusual increase in presumptive Listeria environmentals throughout their facility, and they wanted to kind of reevaluate the methods they were using to confirm, track, and essentially eradicate the Listeria within their facility. After discussing the Listeria PatternAlert assay with them, they decided to give it a shot. So following analysis of 61 presumptive samples, it showed that there was, 90% of those were sharing a single common pattern when run on the Listeria PatternAlert. They also found that it was concentrated to loading areas and employee entrances. So as they continued to analyze this data, looking at the metadata that was provided in their company-specific database, they were eventually able to reveal the root cause. So trucks were actually transporting that Listeria from the farm to the facility's loading areas, their parking areas, and essentially it was further being distributed throughout their facility by their employees walking through those same areas. This was a very interesting case. Company-wide discontinued cultural confirmation, and they started using Listeria PatternAlert as their method of tracking and trending Listeria presumptives. This client also used this data to advocate for funds in order to correct the issue. And then we have another unique one, Company Z. So our final case study, Company Z, they have facilities across the United States. This company came to us really just proactively. They wanted to broaden the depth of their EMP program. They just wanted to be proactive. And so they had heard about Rheonix. They knew that we were onboarding the technology, and they just used it proactively. So they were going to send us their presumptive enrichments anytime that they got a presumptive. They weren't expecting to have this happen. So, after a number of presumptive enrichments were sent over a, specific amount of time, they were actually finding that the same pattern was being found in different facilities in different locations in the United States. So this was strange and interesting and unexpected, but it did help them to really narrow down their search and narrow down their investigation to figure out, what is causing this. So they were able to, think about the things that those different facilities had in common: the people, the equipment, maybe the suppliers, the materials, the ingredients that were coming in and out of those facilities. So this one was really interesting as well. So they'll continue to use this as their, preventative measure to helping to identify the sources of those Listeria contaminations within their facility. Okay, so in conclusion, the Listeria PatternAlert is really a great tool that provides more specific information. It builds context and provides a focused approach to developing corrective actions. Each scenario is different, but these are just 3 examples of how this technology has benefited food manufacturers and assisted with their plans for eradication of the contamination within their facilities. This provides more information than cultural confirmation alone. With culture, the client's going to determine, is it detected or not detected? But with the PatternAlert, they know if there's a correlation between the 2 different hits or more, and can determine, is this the same Listeria in this location as it is in this location? And if so, that helps them with their investigation. So I'm going to hand it over to Bill next from Rheonix, who's going to show you how to use these tools and how to visualize the data.
All right. Thank you, Megan. And thank you again to everybody that's hopped on today. I'm Bill Pitcher, I'm the business director with Rheonix. I'm going to be going through a little bit more about the instrument, the assay, and then some of the analytical capabilities of the software component of our assay. So we've kind of touched on this information a little bit already, but just to really drive the point home, these are the main benefits of the Listeria PatternAlert assay. It works directly from positive enrichment without an isolate being required. It's a fully automated system run on our Encompass Optimum, and it creates actionable, fast results in 7 hours. There's no master public database. Everything's customized to each individual customer. And then that data can help provide rapid, actionable data to help identify root cause of recurring contamination, eliminate harbor sites, reducing contamination risk, and then addressing emergent issues to really move from being on the defensive to the offensive for dealing with Listeria contamination. So a little bit of background on the instrument itself. It is called the Encompass Optimum Workstation. It is a fully automated sample insert system, and we can detect up to 34 targets per sample using our technology. We use 16 of those targets for the Listeria PatternAlert assay specifically. You can run up to 24 samples at a time, and it's designed with simplicity in mind and with a simple and intuitive workflow and interface built directly into the instrument. There's no reagent prep necessary. We do all that in-house for you, and it does have open system capabilities for custom assay development. Taking a little bit deeper dive into what's actually needed to run the instrument, starting at the top right and then moving counterclockwise, we have our card cartridge that is where we facilitate the entire PCR process. CARD stands for chemistry and reagent device, and this does everything. It does lysis, extraction, purification, amplification, and detection. On there you can see the PCR tubes in white, and then everything else is a microfluidic cartridge. Moving down, we have our touchscreen user interface. Again, that's built directly into the system, and it guides the user through every step of the process, and when everything is correct, it lets you know it. We also have our sample rack. Next to that, we have our manifold, and that's going to deliver your positive and negative pressure for the microfluidics, and also houses our thermocycler. Then we have our reagent kit. Again, we do everything in-house for that, and then pipette tips to run the instrument. Everything is moved around using our robotic handler in the middle here. So how does the Listeria PatternAlert assay actually work? Essentially, it's testing for the presence or absence of 15 separate genomic sequences present in Listeria. Each sample is scored on that positive or negative, and then based on that, we create your pattern for each sample or fingerprint for Listeria. Something important to note here is that sequences occur independently across the Listeria genome, and they're not necessarily tied to one species. So we're never telling you if it's L. grayi or inocua or anything that specific, right? We're looking for just these 15 genomic sequences. There are 2 versions of the assay available. One that both of them will give you a pattern or a fingerprint. The first one is also going to look for that confirmatory target for Listeria species, and the second one adds an additional L mono target to it. So whatever you want to look for. The next couple slides are going to go through what that workflow might look like if you want to use the Listeria PatternAlert. And this one specifically is third-party testing, or if you want to use Eurofins. First, you have your initial presumptive positive using a BAX or a VITUS or a VAC gene, whatever you use to get to your presumptive positive. You pipette an aliquot from that presumptive positive enrichment, and then you add in the enriched sample to our Rheonix Easy Lyse Flu Buffer, ship it to Eurofins. They're going to process your samples, and then you will have access to your customized database to filter through the results as you wish. If you wanted to bring this technology in-house, the first part's going to be the same. You get your presumptive positive using whichever method you prefer. You pipette an aliquot from that presumptive positive enrichment. There is a bead beating step because we know that Listeria is pretty tough, and then you transfer that sample to a carrier tube, load everything up on the instrument, hit go, and it is a 6.5 hour assay after that point. Once it's done, you then marry that with our PatternAlert software component, and you get to filter through your customized database. So what do you track, and what does that database sort of look like? So on the top here is what we would classify as pre-run. What do you want to record for each sample prior to running it through the Listeria PatternAlert? There are some fields that are required, but you can add up to anything that you want, and we don't have a limit on that. So in this situation, we have a fictitious company, Hamco, and they're recording facility location 1, a sublocation 2, collection date, screening method, screening date, area, and then zone. And then you can add anything else that you want in there that you want to track and you want to run metrics on. Afterwards, on the bottom here, is the data that's going to be filled in post-run, so after you run it on the instrument. In the highlighted section here, depending on which kit you use, you're going to see your confirmatory for LS or LM, whether it's positive or negative for each one. And then you're going to see the pattern name that is associated with that sample. Every time you run the instrument, you're going to get back your metadata, and then you're going to get all the results associated with each of those samples and metadata, location and date the samples were tested using the assay, your confirmation again for LS or LM pattern name. You're also going to get which individual targets of those 15 genomic sequences tested positive. That's not pictured here. So what do you do with that data after it's generated? Our analytical tools enable users to drill down by pattern frequency, facility, location, sublocation, collection date currently, and we're always adding to that. We're always trying to make this a more actionable and intuitive tool for our customers. Using the same fictitious company, Hamco, we looked at the data that they've been running, and first we sorted by pattern frequency. We looked and we saw that Ham 11, their pattern 11, showed up much more frequently than anything else. So we decided to drill down a little bit more and look, where is Ham 11 actually showing up? Now, this can be filtered further by sublocation. So if we want to look at packaging room, for example, we could filter that out into each of its sublocations and see how often Ham 11 is testing positive there, and then are there any other patterns associated with that location? And this is just a couple of examples of how you can actually filter and use the data. It's, again, completely customizable to you and what you want to track. So as we sort of wrap up here, we talk again, like, what can we use this data for? It can be combined with existing operational data to help QA/QC teams to identify recurring or persistent Listeria in facilities, evaluate how that Listeria is traveling, accelerate your root cause analysis, leading to SSOP adoption changes. And it's also a CSV file in its most basic format, so we can integrate into any standard environmental mapping software. And again, what are those main features that we just talked about? Again, no ISO required, it works directly from positive enrichment. There's no master public database, everything's customized to each individual user. It's a fully automated system, no user intervention after you load up your samples, quick actionable results in 7 hours, and the training infrastructure is very low in terms of what you actually need to run the instrument. So that being said, that's all I have, and thank you again for your time, and I'll turn it back over to Scott for Q&A.
Great, thank you, Bill. Going through, we've got a series of questions. I'd like to welcome our participants back in here. I'll get my cam back on as well. See a few questions then that came in. I think maybe one that was asked by someone really early on. It looks like he's since left, but maybe an appropriate place to start with. What would be the source of Listeria? Where does it come from? And I think it's interesting as we continued on in presentation, we talked about the case studies. I think that illustrated a few examples on where it could come in from, being essentially ubiquitous in the environment. They really come in from anywhere with supplier materials, pallets, employees, trucks, and so on. But just kind of want to start there, and Dan, maybe toss it over to you then, share your thoughts on that if you want to elaborate on that.
Yeah, I mean, I think you hit the nail on the head there, Scott. It's ubiquitous in the environment. So anytime you have soil, any chance for soil to get onto anything, whether it be your shoes, and then work its way into a facility, I mean, that's going to be a really big source of Listeria in a production facility for sure. And then once it gets in, of course, it's it can live basically in many different places that are wet. So.
Okay, great. Next, got a couple of questions here regarding the PatternAlert, the capacity a little bit. I can't recall if you mentioned this, Bill, in your conversation, but what is the sample capacity of the instrument?
Yep, so each run you can run up to 24 samples.
Excellent. Thanks for adding that. And then I think you mentioned this, but let's reiterate it a little bit. How many Listeria species provide a positive detection? So what can it detect, I suppose?
Yeah, so we did touch upon it a little bit, but we're never actually determining if it is one specific Listeria species. We're looking at 15 predetermined genomic sequences to assign a pattern to each sample tested.
Excellent. Yeah, thank you.
I think the question, I think maybe I could add to that. I think the question maybe the person is alluding to is of the newer Listeria species, you know, will the Rheonix system actually see those newer species and see them as Listeria? I think we know the answer is yes for many of them. I don't know that we know for all of them, I guess would be what I would say to that. But I think that's true of any rapid screening method out there right now, currently.
Yeah. Okay. I guess continuing on a little bit with you, Bill, what are your experiences with false positives with the pattern alert assay?
Yeah, absolutely. So during our initial validation of the method, we actually ran 100 known negative real world samples and had no false positive results, no positive results from that. 200 of which were actually fully culture confirmed as negative.
Excellent. Okay, and then as far as certification, I know Dan, you kind of alluded to it a little bit. Does the pattern alert method have AOAC certification? You guys working towards that? If not, can you update a little on the status of that, Bill?
Yeah, so we don't currently have AOAC approval for this, but most strain typing methods do not require AOAC or third-party certification. Some customers have expressed interest in having certification for use as a confirmation method, and Rheonix is investigating that. Our internal studies have indicated that the method is fit for purpose for non-culture based confirmation.
Excellent. Yeah. Okay. And then maybe on the lab side, a question came in here regarding lab certification. So does the lab need to be ISO certified to use the technology? Not sure who would be most appropriate, Megan. I mean, we are ISO
So I'm not
certified.
sure if it's needed to use the technology, but we are ISO certified. So Bill, do you know the answer to that?
I'm sorry, could you say the question again?
Yeah, the question is regarding the lab using the technology. Does that lab using that technology need to be ISO certified?
I'm not entirely sure of the answer to that. I'll have to follow up.
Well,
Okay.
one thing I can say, yeah, I would say it's no, but I think anybody could use it. But I do think, yeah, you would prefer an ISO certified lab, obviously, be running your testing for you. But also any laboratory that's going to use the method, as with any method you bring into your laboratory, you're going to want to verify that your laboratory can actually execute the method, and there's specific requirements to do those verification studies.
Yeah. Thanks, Dan.
Sure.
Okay, I guess continuing on a little bit with the lab side. Megan, you talked a lot about using pattern alert. We talked a lot about cultural confirmation. Can you perform both cultural confirmation and run pattern alert at the same time?
Yeah, so you definitely can. And this is on a client by client choice basis. So if they want both methods ran, we can definitely do that. And then both methods and results would be reported.
I can add, I can say to that specific question, and I did not share any of the data here, but we do have a good amount of data, I would say, that's showing really excellent correlation between the results we're seeing between culture and the pattern alert method.
Okay. Excellent. So I got a couple of questions here on availability within Eurofins. One question here, is this available in Eurofins labs in Canada? And as Megan alluded, currently the Minnesota lab is the one performing that. I would believe if we have labs in Canada, we can always ship them over to Minnesota. But I don't know, Megan, can you kind of elaborate, confirm?
Yeah, so currently the Mounds View location is the only location running the pattern alert assay. However, we do have a process set up internally with all of our labs. So no matter what lab you're submitting to within the Eurofins network, or even if you're submitting to a different laboratory or you have your own internal lab, we can have we have a process for how you can ship enrichments to us, and we can still run the method for you here.
Excellent. Another question kind of along the same lines. This client here uses Eurofins currently to perform Salmonella Listeria testing. Is this something that we can just ask to get that presumptive notification from them?
Yep, so we would just ask that you work with your local Eurofins contact, and they can reach out to me and we can get it all set up for you. It's really a simple change on the account, just a few changes to get it set up and going, and we'll probably do, like to do introductory call, making sure that we have everything covered before we get started, but it's really a pretty easy process.
And important to point out, I know you said Salmonella, but Salmonella is not available on the Rheonix platform. I'll let Rheonix comment on their thoughts there, but as of now, this is a Listeria only technology.
Yep. In terms of Salmonella, we're definitely aware of a need for some type of assay, but we're trying to figure out exactly what that looks right now in terms of serotyping. So we're still evaluating that need.
Yeah, another question kind of along the lines of some of our earlier questions, but it points out the EU market requires ISO validation to use the Rheonix for confirmation. And question is, are we heading towards that? So,
I mean, I can say my opinion here. Yes, we are. I think we are heading that way. I think one of the issues, at least from the U.S. perspective and AOAC, is while there is a methodology to do AOC validations of confirmation methods, it's evolving. Let's say that in the discussion is actively evolving about the appropriateness of that and other ways and how we might actually validate molecular confirmations. So it's a new area, as I mentioned, and there still is, I would say, TBD. There's still a lot we need to learn.
excellent. New question just coming in. When doing verification of this technology, is it only for environmental monitoring samples or for food samples too? Maybe kind of elaborate on any matrices and so on from there.
So I think initially, and correct me if I'm wrong, Bill, but I think initially this was aimed towards environmental monitoring, but we can absolutely run it on product samples as well. And I think there can be some interesting correlation, especially if there is some contamination going on in the environment and you end up with, presumptive product. It would be very interesting to be able to correlate whether or not that's the same pattern going from environment to product. So it is kind of geared towards an environmental monitoring tool, but it can transition over to evaluating product as well for patterns and to see if there's that correlation.
And I can say from the lab perspective, we're seeing, and we have seen in recent years, a big increase in the amount of people doing species testing of product and moving away from mono, whereas previously that was really only something you saw in environmental testing.
Great. Okay, let's see here. This question here, I believe, is regarding, let's see here. Can it be said that there is a lower chance of presumptive samples to not detect with Rheonix? So we're seeing an area with 9 presumptive results that have all gone to not detect with no confirmed positive results. Can we kind of talk a little bit about that a little bit?
Yeah, I think the question is that they're asking is this platform less sensitive, perhaps, than culture as a confirmation tool? Maybe that's what they're asking, or they're asking, is it less sensitive than the primary screening method, which is typically a PCR method? And I think the answer is unknown, or it's going to be a variable depending on the situation and depending on the given matrix and different factors. So it's a good question, and I don't know that we necessarily know the answer. All I can tell you is that, again, as I mentioned already, the data so far, the correlations between culture results and Rheonix when used as a confirmation tool are strong. So it's looking like that sensitivity difference, if it's there, is very minor.
Okay, that's a good response. And I believe that kind of hits most of our questions. Don't look like any more are coming in, so I think we should go ahead and wrap that up here. I'd like to thank you all then for attending. I'd like to thank Megan, Dan, and Bill, especially Bill joining us from Rheonix. It's a very pleasure to have you on here and talk a little bit about the Rheonix platform. It's excellent technology to share with us all.
Thanks, Bill.
So Yeah.
Hey,
So I
can
think-
I just say, Scott, quickly that the Eurofins team has been an absolute pleasure to work with, and if you do need a third-party testing lab, absolutely recommend them. So
Thanks, Bill.
Yeah, thanks.
Thank you.
And then one last thing, I just want to remind everybody, we'll of course have a recording of this presentation along with a copy of the slides sent out within 3 business days. So I guess that wraps up our presentation, our conversation today. Thanks you all for attending. We'll see you
Thanks,
next
everybody.
time.
Take care.
Thank you.
Bye.
Thanks, everybody.